Obviously, we are good to go. Streaming YouTube also now.
Yeah.
ஆமா பக்கத்து ரூம்கார தான் சேர தூக்கி தூக்கி அவரு சொல்லி வைக்க முடியாது கோட்டா கோட்டு நல்லா இருக்கா சும்மா அவ்வளவுதானே நீங்க வந்துட்டு தான் பாக்க சொல்ற இன்னும் நீ எப்படி சொல்லிட்ட அவனுக்கு பையனுக்கு ஸ்டேட்டஸ்
Hello everyone.
Hello, ma'am.
So we are waiting for some remaining participants, and now we are starting our today's day. Good morning, good afternoon, and good evening to everyone. Greetings from Gujarat, India. I am Bhagyashree Dangar, Junior Research Assistant at the Center of Excellence for Wildlife and Conservation Studies, Bhaktakavi Narsi Mehta University, Junagadh, Gujarat. I am working on Bryophytes of Girnar Wildlife Sanctuary, and I am also a proud member of IUCN SSC Bryophyte Specialist Group. It is my pleasure to welcome you all in event of World Bryophyte Day 2024. This is a special day as it brings together biologists and nature enthusiasts at the outset. I am pleased to share that from 18 different countries and 26 states of India, the participants are participate for this day of World Bryophyte Day. What a remarkable gathering of all bryophyte lovers. We are here today to discuss the incredible world of bryophytes. So, bryophytes are tiny plants that play a huge role in our ecosystem. Today's talk will showcase leading experts from both India as well as UK. Making this special day, we are able to a truly international events. Now, I'd like to invite Dr. Rajesh Ravia, Associate Professor of Botany at Bhaktakavi Narsi Mehta University, Junagadh, and our esteemed member of our advisory committee to provide an introduction to today's event. So please, sir. Unmute your mic.
Hello, everyone. It is an honor to be here to celebrate World Bryophyte Day. The day is dedicated to recognize, appreciate one of the most ancient fascinating group plant is our bryophytes. Bryophytes are more than just small, but overlike the plants, they are pioneers in our ecosystems. Then recovery regulators, the water cycles, provide habitat for the various organisms. The creep arrows, the fight against the climate change. This is the one of the most crucial in this point is climate change. And their conservation is critical not only preserving biodiversity, but also for ensuring the stability and the health of ecosystem changing the climate. So as we celebrate World Bryophyte Day, let us commit to raising awareness about the importance of these plants, supporting our conservation efforts and the promoting research can look their full potential addressing environment challenges. I'm heartily welcome to our all dignitaries, Manju madams and sirs and Bhageshi and all participants. So this is a very good platform for this our conservations and the knowledge regarding the bryophytes. So thank you very much, Bhagyashree. Please carry on.
Thank you so much, sir. So now the time for our first speaker. So it is my honor to welcome and introduce our first esteemed speaker, Dr. Manju C. Nair, Associate Professor of Botany at the University of Calicut, Kerala. Dr. Nair is one of India's most respected biologists with an exceptional body of work spanning bryophyte taxonomy, ecology, and molecular biology. Her contribution to this field have been nothing short of remarkable. Dr. Nair has discovered more than 16 new species of bryophyte, shedding light on previously unknown aspects of plant diversity. She has successfully completed over 5 major projects has delivered more than 23 expert talks at national and international platforms, and her work has been widely recognized with over 105 research articles published in prestigious national and international journals. In addition, she served as a reviewer for several leading journals on bryophytes and has presented over 22 research papers in various conferences, seminars, and symposia. Her expertise has earned her 6 prestigious national and international awards, and she has mentored over 6 PhD students, guiding them to academic success. Dr. Nayar's dedication to bryology is evident not only through her research but also in her commitment to education and knowledge sharing. We are fortunate to have such a distinguished expert in bryology with us today. It's an absolute pleasure to welcome Dr. Manju. Today, she will talk on patterns of distribution of bryophytes in Western Ghat. Without further delay, I am handing over this virtual stage to her. Please, madam, carry on the lecture.
Can you hear me?
Yes, ma'am.
Okay. Good morning, good afternoon, and good evening to one and all. So I think nearly 18 countries. The participants are from various time zone, and I think it's a pleasure and honor to me to attend this meeting. I thank Dr. Rajesh sir and Bhagyasri. So I think Bhagyasri is usually communicating with me, and I think she is a dedicated researcher. And I thank all the members, those who have contributed for this day, especially on the Bryophyte Day, World Bryophyte Day. So I think it is first time we are celebrating here, isn't it?
Yes, ma'am.
Yeah. That I congratulate you people for suggesting or selecting this day to evaluate our Bryophyte studies in Western Ghats. So can I share my...
Yes, ma'am, please.
Is it shared?
Yes, ma'am.
Okay, today I'm planning to give some ideas on the patterns of distribution of bryophytes in the Western Ghats. So just there is, I think, more than 150 slides, but without any write-ups. So only photographs. So you can view the diversity of bryophytes in our area, but it is least studied. You can see from some slides that our studies are very meager compared to other countries. So I am myself Manju, and I am working as Associate Professor of Botany in the Bryology Division of Calicut University in Kerala. So earlier I worked in a nearby college, the Samurins Gurayappan College, for more than 10 years. After that, I came here for last 3 years. So it's a proud moment for me to address you all. Thank you all. And I am coming to my topic. So you all know that bryophytes are have been divided into 3 classes. That is, now it is elevated to the 3 divisions, that is Microhabitats, Anthocerotophyta, and Bryophyta. And we will say the liverworts, hornworts, and mosses for easy recognition. And this is the distinction of species. And the major characters of the bryophytes are we can say that they are the amphibians of the plant kingdom. Non-flowering, non-fruiting, and non-tracheophytes, and the gametophyte is prominent, and the sporophyte is parasitic on the gametophyte. So here you all know that the gametophyte is from here in Western Ghats. The rainy season starts from June and it ends during October, November. And up to that period, the gametophyte is very live. And along with this gametophyte, some sporophyte will come from June itself up to December to January of next year. So that depends upon the species. And we all know that there is no flower, no fruiting, but we can say that there is a flower in Bryophytes, and we will say the flowering of Bryophytes as the cluster of Antheridia. So other characters, they are considered as non-tracheophytes. So we all know that they are regarded as non-vascular plants, but there is vasculature. So the xylem-like cells and phloem-like cells are present in each group for the conduction of water and minerals. So there is some structures like xylem and phloem. That's why now the term is redefined as non-tracheophytes. So we can say now onwards, they are the non-tracheophytes, not the non-vascular plants. There is vasculature because some plants that starts from 0.2 munum to 60 centimeters long. So that 60 centimeter long plants, and some even show some hanging plants, they are more than 1 meter long. So they also need water up to the top of the plant. So there is some mechanism for the conduction of water and minerals. That's why it is referred to as non-tracheophytes. So coming to our distribution pattern. So we can say that the world Bryophytes, it is around 25,000 next to angiosperms. And in that itself, the mosses are the most species group with 18,000 species. And in India, we have nearly 3,500 species described. And in Western Ghats, 1,229 species. But we are expecting more than 2,000 species. This is a small outline. And considering the different states of Western Ghats, that is Tamil Nadu, Kerala, Karnataka, Goa, Maharashtra, and Gujarat. So from this graph, it is clear that the Tamil Nadu and Kerala is in top portion. That is only because that the diverse microhabitats and macrohabitats, the vegetation types are very much distributed in Karnataka, Kerala, and Tamil Nadu. But the studies are very meager in Karnataka. That is why the number is very below 450 species. And in Kerala, there are about 750 species, and in Tamil Nadu, 840 species. So we can see from the graph, the mosses are the most species group in our region also. In Gujarat also, it's nearly 50 species they have described, and more than 50 species from Gujarat, but the Western Ghats region starts from Tapti River. That Tapti River holds nearly this much species, 33 species of mosses and 2 hornworts and 14 species of liveworts. And it is based on the literature study. And Maharashtra also very few studies, only 1 or 2 are have done some major works on Maharashtra. And Goa also only 1 work is there. And we have also documented some species. But in Goa, you all know that the forest area is very big or very low in that area. But there are wildlife sanctuaries and national parks. So this is based on our collections and earlier reports. So coming to the various microhabitats, habitats of Western Ghats, we can divide based on the occurrence of this vegetation. I have divided based on my experience in the field during the last 25 years. That is tropical dry deciduous forest, tropical moist deciduous forest, and tropical thorn forest. We can club together in a single group. Here, very few mosses and liverworts like Rixia, Cyatodium, etc. are seen, but we can see that the species growing in this area, because this area is very dry, and the species growing in this area never seen on other areas. And the next club is tropical semi-evergreen forest, tropical evergreen forest, montane wet temperate forest. Here we can see a good number of species, mosses, liverbirds, and hornbirds. Nearly 12 species of hornbirds are reported from here, and this diversity is we cannot imagine, and this is based on the gradation in the altitude also. The diversity may differ based on the altitude also. The next patterns, southern subtropical savannas and montane wet grasslands. So the montane wet grasslands are associated with the montane wet temperate forest. But here the species occurrence is very low. Very few species are seen in this area because a direct light is approaching in this area, and we can see only very few mosses and very few leafy loverbirds, but not common in other areas. The species growing in this area never grow on other areas. So another vegetation pattern is wetlands and riparian forests. That is, we can see some thalloid and leafy liverbirds, especially leafy liverbirds we can see from these areas. So next pattern is croplands and agricultural lands. So our croplands and agricultural lands, that is teak plantation, then the tea plantations, coffee plantations, that means in that area there are several mosses and leafy liverbirds. The diversity is very less in this area, but the frequency of occurrence of species is very high. And next, we cannot say this is vegetation type, but it is a habitat, that is around human habitations. We can see several species in and around our homesteads. Very few mosses and liverbirds, and we can see some hornbirds in the low altitude regions of. There are also some there are shady regions and open land we can see around human habitation. So these are the major vegetation division based on our Bryophyte collections in Western Ghats. So we can see some photographs, next slide, some photographs of this different forest and which are the species occurring in that area. So this graph also shows the distribution of bryophytes along the major vegetation types. So you can see that from tropical thorn forest and tropical dry deciduous forest are the vegetations with a very less number of species compared to the montane wet temperate forest and the evergreen forest. So we can see that the montane wet temperate forest and evergreen forest always show some high degree of occurrence of bryophytes, especially the mosses and the liverbirds. So, and next to montane wet temperate and evergreen forest, we can see semi-evergreen forest also shows some pattern of development of the Rexia, the Bryophyte species. But because this area shows a mixture of moist deciduous trees and the evergreen trees. So, next, the croplands and wetland and tropical moist deciduous forest shows a maximum diverse species. The low vegetation is seen in the thorn forest, dry deciduous forest, and montane wet grasslands. So the first club, the tropical dry deciduous forest, tropical moist deciduous forest, and tropical thorn forest. So this is one of the photographs from Junagadh Wildlife Sanctuary. This is the pattern. This is the aerial view of tropical dry deciduous forest. During the most of the years, the area is with a dry nature. So I think there is a rainy season only once in a while within 2 years. So we can imagine what all species will be there and how this species is distributed in tropical dry deciduous forest. So this is also another photograph. This is the most deciduous forest. Here also, most of the time, the epiphytic habitat and the above ground area also will be dry in nature. So this is also a moist deciduous forest in Wayanad Wildlife Sanctuary. So this is a scrub jungle in dry deciduous forest of Attapadi area. So you can see the thorn forest. So in dry deciduous forest itself, we can see a patch of thorn forest. But you see the microhabitat here. So you can see a special type of bryophytes from this area. So you see this. This is the dry, hot month in our area. That is nearly December, January time, we have collected the species. This is a rare species, that is Exomothica ceylonensis in the lower portion. You can see. So this plant is with the sporophyte. So you see in hot condition also, this species produce sporogonium. In the upper case, you can see one Selaginella vitae. This is a resurrection plant. And along with that, we can see one Rixia species, that is Rixia ciliata. So this species also, this is greenery in nature. So we can see that there are several adaptations to thrive these plants in that area. So you can see this is a cilia. Cilia will be developed in each plant, that is Rixia ciliata, especially ciliata. Some species show some adaptive characters to thrive in that dry conditions. Some may develop the scales, full of scales will be covered in the thalloid region. So these plants produce porogonium, but the most cases are reproduced by vegetative means. This is another species, the Rixia growlei. This is also growing in dry habitats in Attapadi area and the dry Indira Gandhi Wildlife Sanctuary of Tamil Nadu area. So you all know that the area is very dry during most of the time in a year. So you can see the scales in that area. So the species produce porogonium as well as it is protected by some other means, the scales, cilia, and other structures. And the thallus is very stiff in nature. So this is also an adaptive characters. So, but the species occurrence is very less in this area. So we can see this Bryon coronatum, Hyphla involuta in and around all areas in Tamil Nadu, Karnataka, Goa, everywhere we can see this plant. It is mostly found in and around our homesteads and in dry condition, that is in dry deciduous forest and moist deciduous forest also, the species is highly distributed. So next pattern is tropical semi-evergreen forest, tropical evergreen forest, montane wet temperate forest. So you all know that the evergreen patches. So we will say the evergreen patches normally, just example is Silent Valley National Park. So the Silent Valley National Park is always, it is greenish in nature. So there is a patch of montane wet temperate forest in the Silent Valley itself. So, but except that area, poor area, all other areas with a tropical moist, evergreen species. So in this area, the diversity of species is entirely different from the first club, that is the moist deciduous and dry deciduous species. This is also the Agasthyamala Biosphere Reserve in Trivandrum district of Kerala, and this is the Jogfall area of Karnataka. These are all evergreen patches. Sahasringlingania jog in Karnataka. So this is one wildlife sanctuary in Goa. That is, it is a semi-evergreen patch. But here also, the it is a semi-evergreen forest, but the forest is open compared to other areas. This is also from Goa, Goa Wildlife Sanctuary. So you can see some species inside that area, Mahavir National Park in the Goa area. So in Goa, there are 2 national parks and 6 wildlife sanctuaries. Apart from the beach areas. So this area, we have collected nearly 40 species of Bryophytes from the Goa region. So this is the montane wet temperate forest of high altitude regions. We can see this type of forest is most common in the South India, especially in the Irvigulam National Park, then Kodaikanal, then Ooty, then in Kerala itself there is in Wayanad, we can see a patch. In Salem Valley also we can see one patch. The Irvigulam National Park is famous for the Nilagiri tar, that is the endemic goat in our area. So we can see some patches of forest inside the grasslands. This is the montane wet grassland and montane wet temperate forest. So this is Anamudi, south of Himalayas. There is the highest peak, 2,695 meters above sea level. And this is another the forest path, that is the Shola forest. It is termed as Shola forest, South Mountain Wet Temperate Forest. It's also near to our area, Karimbuja Wildlife Sanctuary. These are all patches of forest. So you can see thick growth of bryophytes in this forest. So this is another view from Wayanad. This is from Kannur district. And this is the patch of forest found in Silent Valley. So only one patch is with a mountain wet temperate forest. And you see the inside forest, the evergreen, semi-evergreen, and mountain wet temperate forest, we can see this type of diversity. So from base to top, we can see, so we cannot say this is a single plant. It may be more than 100 species we can collect from this plant. So that from top, base to top, this is the inside forest of Shola region. So hanging from the branches. So this is also the hanging from the branches. This is, I have told you, it's more than 1 meter in length, and it needs water and minerals for their survival. That's why it is known as the non-tracheophytes. They need xylem-like cells and phloem-like cells in these cells. When we take this section, we can see that there are certain cells, that is hydroids and leptoids are seen in these plants. So this is the inside patches, evergreen forest we can see. This is with the full of Cyatodium cavernarum. So this is Physidens species growing, Bryoides, Physidens bryoides. This is also another national park, that is Cyatodium cavernarum is very much growing in the epiphytic habitat. So this is a common view in the mountain wet temperate forest. So how can you count this species? So we can see Astrella, Pogonatum, then Putium, then some other algal growth are seen. This is a small area with more than 5 species. So some leafy liverworts are also included in this species. So from one patch, we can see more than 5 to 10 species. So this is Markantia polymorpha. It is very common in the high altitude region. Only we can see Markantia polymorpha only about 2000 meters height. So Gaucheria Schizopleura, this is a very rare species in the high altitude region. So this is a leafy liverwort, but you can see some other species, Pogonatum and Campylopus also growing along with the species. So Imbrium alpinum, this is alpine species. So earlier it is known as Bryum alpinum. The name has been changed to Imbrium. So Hypopterygium tamaris, it's a very common species in the high altitude region, but it is found only in the mountain wet temperate region. We cannot see this species in the evergreen forest. So this is also Rhodobryum. There are several species of Rhodobryum. One is Rhodobryum oberti, Rhodobryum giganteum. All these are growing in the high altitude evergreen and mountain wet temperate forest. So we can see that one tree is here. So what we see depends on how we view it, how we view this plant. So we can say that an angiosperm taxon will say that, oh, this is a tree. So, but myself and my students and those who are loving the Bryophytes, they will say, so there are a good number of species. So from base to topex, this is around 30 meters girth plant. And we will imagine more than 30 meters. So in the evergreen areas, we can see nearly 1 meter to 2 meter girth plants. So from base to top, we can see nearly 15 species. One MSc dissertation is enough with this plant, one epiphytic plant. So this is the diversity. With the one tree species, we can see that nearly 15 species we can collect from this plant. So how you imagine that, how our diversity is, or how the Bryophytes is diverse in our area. From the first figure, we have seen that the Tamil Nadu and Kerala is far better than other states, because the studies are started during 2000 itself in Tamil Nadu and Kerala, but it's very slow in other states. So that's why So we have only very few species. We are saying that our studies are very meager compared to other countries. So next pattern is southern subtropical savannas. It is found only in some parts of Nilgiris, Annamalais, and Palni hills of Tamil Nadu and Kerala. And mountain wet grasslands, it is found. I have seen that. I have shown you the mountain wet temperate forest. Along with that, we can see the grassland. That is the mountain wet grasslands. So this area is very cool. Wet condition is there, but with intense sunlight. So the direct sunlight will be given to this area. So the diversity of the species is entirely different from that area. So this is the subtropical savanna area. This patch is the mountain wet grasslands. So the grass species also different from the normal grassland areas of low altitude areas. So we can see this patch. So this plant, the Campylopus, mostly this Campylopus is seen only in the mountain wet grasslands. So this is an iconic species of mountain wet grasslands. You can see some grasses in between the Campylopus species, and some may mistook the species as sphagnum. So in South India, there is no sphagnum. In northern region, that is in Himalayas and Assam, that area, we can see some sphagnum species. But in South India, there is no report of sphagnum. But the nursery people may sell this species, this Campylopus species as sphagnum. So this is another species, which is Campylopus species, which is growing in the wet mountain grasslands. And this is also another species of Campylopus, which is also growing in the wet mountain temperate forest. Along with that, you can see some sporogonium here. This is Centostodon species, along with the Campylopus. I will show you the next species. The Pogonatum. Pogonatum is there are several species, nearly 15 species of Pogonatum in our area. That Pogonatum also growing widely in the open land area. They need light, sunlight. So this is the Endostodon species, which is growing along with the Campylopus. And this species, this Endostodon, is found only in the grassland area, which never grow along the sides of the mountain wet shola forest and other evergreen forest areas. So you all know the Phenology hygrometrica. We can see Phenology hygrometrica in the open land and evergreen area, semi-evergreen area, and the mountain wet grasslands. But it is very common in the mountain wet grasslands. So you can see this is an open land area, and on the cuttings we can see this Phenology hygrometrica along with the Bryum argentium. So next pattern is wetlands and riparian forest. In wetlands and riparian forest, we can see that the sites of riparian habitats. It's full of leafy liverworts and thalloid liverworts like Astrella, Dumotera, Targiona, everything we can see from the riparian habitat. And the other character of riparian habitat is the epiphyllous habitat. Epiphyllous species are seen in riparian habitat. So this we can see as a bed. So we can collect the specimen as such from this. So this is not a single species. When we collect this and see under microscope, we can see more than 3 species we can see from this bed. So this is the aquatic species, Rixia fluitans. So it's also found in the evergreen riverine areas. And this is our new species. It is collected from this, the dam site area. This is a marshy area. This is Rixia. It's very interesting. And this species shows the upper photosynthetic region on the lower part. So this is another species, Micrometrium varanicum. This is also our new species. This is also growing on the marshy land area. So there are several species growing as aquatic and on the sites of riparian habitats on the marshy land. So the Rixia fluitans also growing on the aquatic habitat. It's float on the habit, and when the water resides, it grow in the marshy land. As the thallus may the size of the thallus may differ in that species. So next is the croplands and agricultural lands. In croplands and agricultural land also, so we can see a number of species. So this type of terrain, we can see in Ooty, Kodaikanal, Munnar. So all high altitude areas, we have tea plantations and coffee plantations, then rubber plantations, etc. So in all these areas, the coffee plantation and tea plantation is well versed with the leafy liverbirds. So we can see epiphytic habitat is, this is another photograph. You see this. So now all the tea plants and all the coffee plants, we can see this type of microhabitats. So this type of distribution in that area. And there are very rare species occurring in this microhabitat. So this is another view from a, this is coffee plant. So these are the distribution. I have shown you only very few photographs. So there are several photographs, but to give awareness, so what type of vegetation types we have and what all species distributed in that area, how the pattern is different. So in evergreen, we have seen that the evergreen and moist mountain wet temperate forest are the diverse areas. But the diversity of each species is seen in some other areas also. In human settlement also, we can see some species. It is diverse in that respect. So coming to next we are coming to the habitats and bryophytes. What are the different microhabitats and bryophytes associations? So we can broadly divide the microhabitats as epiphytes, terrestrial, and aquatic species. So the epiphytic habitat also we can divide this. We can diagrammatically represent one tree, and we can divide the tree into different zones. We can divide it into 5 different zones: zone 1, zone 2, zone 3, zone 4, and zone 5. So the demarcation we can see that here, and we can see that the distribution of each species or the growth of species is different in different microzones. So the buttresses, the base of trees, buttresses and shrubs up to 1 to 2 meters length, you can see some species. The lower part of trunk. So in the earlier photographs, that 30-meter girth plant, I have shown you some 15 species. In a single, up to the zone 3 area, I have shown the 15 species. The zone 4 and zone 5 is missing in that area because that tree is found in the open land. So we can see the zone 4 and zone 5 only in the evergreen and moist mountain wet temperate forest areas. So we cannot see the zone 4 and the zone 5 in moist deciduous, dry deciduous, and open land areas. So the next zone is lower part of trunk, upper part of trunk, branches and twigs. Then the epiphyllus. Epiphyllus also we can divide into live leaves and dead leaves. Then rhizome of ferns, logs, that is decomposing wood. The association of species in each zone is different. So you can see this is the buttresses area of a tree. And this is, next photo is the above buttress, that is the bark of the tree. The first photograph, first right photograph, and second photograph is in a single plant. And the first one is from, the first photograph is from tea plantation. So you see the bark area is with a good number of species, and it is diverse in habitat, and it has a good lighting condition in this area. So that's why the species is diverse in this area. See the bark with the cyatodium. So this is also in bark area. So this is in bark and branches. From base to top, we can see some species. Maybe it is one species distributed in a single area, but up to from the top portion, we can see some other species. So this photograph you have seen, this is also, we can divide from base, middle, and top portions. So this is the top portion hanging from branches. So this is the zone 5 area. This is the hanging species, and some ticks maybe we can collect from the basal portion. So that also may be different. So the roots of higher plants. This is a myristica swamp region. So here also we can see some species. So on logs, the species growing on the logs never grow on the live trees. This may be different. So in some cases, we can see some species intermingling with the logs and epiphytic habitat. But most of the cases, the logs and the live bark are different. So epiphyllous habitats. This is a very common feature in the riverine areas. So you can see here 2-3 species of epiphyllous habitat. This is mostly in the Leguminaceae members. And some mosses also grows on the epiphyllous habitat. So coming to next habitat, that is the terrestrial habitat. We can divide soil and rocks, wet soil and dry soil, the land cuttings and flat areas. Rocks also we can divide big and small rocks, wet and dry, big wet and dry, and small also wet and dry. Other rocky structures, concrete walls. We can see that the concrete walls are full of with the bryophytes like Bryum coronatum, Hyophylla involuta, etc. So this is the pattern. So we can see some pebbles. So that the species growing on the pebbles never grow on the large rocky patches. This is the land cuttings. So you can see Astrella, all Astrella species. So this is convert, that is the Anthosaurus angustus growing on the cuttings. This is Astrella, the male receptacle of Astrella. You can see the male thallus of Astrella, Khasiana. So this is Bryum argentium. So this is a different microhabitat. So this area is full of pollutants. So creeks and crevices of rocks, we can see some species of physidans, Anthosaurus, etc. in the creeks and crevices of rocks, because mostly the plant need light, but here we can see only few. That's why we can see only few species of Cyatodium, Anthosaurus, and physidans in this area. We can see from termite mind only very 2 or 3 species they had reported. So we have reported only Physidans ceylonensis from this area. So the rocky patch, we can see a good number of species growing on the rocky patch. This is also rocky patch. 2 species we can see and in a single clump. So next, coming to aquatic habitat. So aquatic, we can divide stagnant and running water. Stagnant, again, we can divide it into floating and submerged. So in stagnant water, submerged rocks possess several species of this vesicularia. Vesicularia aquarium species, or it is used in the aquarium trade. So running water, we can divide into floating and submerged type. So we can see this Rixia floatans. This is seen on the floating habitat. This is Rixiocarpus natans. This species is found only in the pond, that is the still water. So this is Rixiocarpus natans. It's a very rare species. It is found only in South India from 2 or 3 localities, because our the water sources are dwindling day by day. So the species is very less in our area. So you can see that this Rixiocarpus is growing along with the this azolla of fern. So that is the microhabitat distribution in our area. So coming to next to the Bryophyte distribution based on the altitude of Kerala. So from coast to crest, that is from low land, or here in our area, it is nearly 20 to 60 meters height to 2,695 meters. The species diversity is very less. So you can I will show you some species, some low altitude species. This is Octoblepharum albinum. It is known as whitish moss because it possesses leucocyst cells. That is why it looks white in color. But it is when it is wet, it become green in color. So Bryum coronatum, it is very common in the low altitude and the medium altitude area also. We can see the species. So Hyophylla involuta, very common in the low altitude and the high altitude also, but it is very common in low altitude areas. This Gymnostomia lavernicosa, you see the microhabitat. It is very dry, but the species is very greenish. So it can absorb the calcium rich soil, or it can live in the calcium rich soils. So it has the capacity to live in that area you all heard about that. Some species are calcium lovers. The Scopula phylocatracta, that is a calcium lover species. This is also, it's growing mostly in the painted walls or this type of habitat. So Physidum ceylonensis, it is a very common species in the low altitude area. Rexia, there are several species of Rexia. In Kerala, there are nearly 23 species of Rexia. And in Western Ghats, there are 30 species of Rexia. And most of them are low altitude species. The Rexia we cannot see in the forest. Inside the forest, we cannot see Rexia. But in the margin, open land area, or in the disturbed area, we can see Rexia. So this is also another interesting species of Rexia. It's also growing in the low altitude area. Cythodium carbonarum is a very interesting species. It is a fluorescent greenish species. It can grow even any microhabitat it can grow. But it's very common. It even grow in the epiphytic habitat also. So up to 900 meters, 900 to 1000 meters, we can see this species. But it is very common in the low altitude area. Pallavicina, lightly we can see in the high altitude and low altitude area. In low altitude area, there are sacred groves. So this species need shady habitat. In sacred groves, it is very common. So there we can see the shady microhabitats. So Anthosaurus angustus, it's a very common in the low altitude areas. So Nothothylus liveri. So in and around our home itself, we can see the Nothothylus. So these are the common species, and from our survey, we 60 species of Bryophytes are found in the low altitude areas. And we have, I told you, 1,229 species reported from, only reported species reported from Western Ghats. So that other species, that is apart from 60 species, all other species are found in the high altitude. From 500 meter above, we can see several species. So this is Astrella veliciana. This is seen only in the high altitude evergreen forest near, mostly in the land cutting area. So Astrella leptophila. So Rebolia hemisphaerica. This is also found only in the high altitude regions. Plagiocasma appendiculum. So this is a high altitude species, but we can see some areas that's near to Silent Valley area. We can see the species growing on the low altitude area. So I have observed this in the low altitude areas also. So Linlaria cruciata, it's found only in the Munnar, Wayanad, Kodaikanal, then Ooty, that area. So you can see the half moon shaped Jamecup here, Linlaria cruciata. The Dimortira hirsuta, this is the largest liverwort found in our area, in Western Ghats area. So you can see different microhabitat, different nature of the habitat in this photograph. So the Targione hyperphylla, this possesses the sporogonium on the ventral surface. It's also found only in the high altitude region. Pallavicina ambigua, this is found only in the high altitude area, but Pallavicina laeli is found in low and high altitude areas. So Porolla parotitiana, Porolla we can see only in the high altitude area. We cannot see Porolla in the low altitude areas. This is Porolla campylophylla. It's a large plant. We can see from our naked eye. So Anthosaurus crispus. This is Anthosaurus angustus is found in the low altitude area, but Anthosaurus crispus is found in mostly in the high altitude areas. Pogonatum microstopum, I have shown you. Pogonatum normally found in the high altitude areas. It never grows in the low altitude areas naturally. So Campylopus is found only in the montane wet temperate forest. It is never found in the evergreen forest also. It's found only in the montane wet temperate grasslands. But there are several species of Campylopus in our area. Campylopus fragilis. So then Bryum argentium. Bryum argentium also, it's growing in the high altitude area and medium altitude. It is never found in the low altitude areas. Low altitude means 100 meters below, it is never found. So this is the species distribution, that is 1,229 species. It is with 827 species of mosses, 20 species of hornbirds, and 382 species of liverworts we have identified. So I told you the estimated diversity of our area is near more than 2000 on 2000, above that. We can say that in Kerala we have estimated 2000 species. In Tamil Nadu also likewise 2000 species. In Karnataka also 1500 or more species we can collect. So this is the trend of Western Ghats studies, that is from the mass studies by Vohra et al. In Silent Valley, 83 species to 1,229 species in 2024. So this is some new distributional records. Nearly 6 species to Asia, to India 26, South India 48, Kerala 97. We have 85 species of endemic species. A new taxa, including Dr. Dulip Daniel's report, that is 35 species. We have reported now 20 species and 4 species in edit. So it is about to publish, and Dr. Dulip Daniels reported one new genus from Silent Valley National Park, that is Indoputia santeri. The group also doing a good number of work in Kerala and in Tamil Nadu also. So we have. So this is, so you all know that the studies on the angiosperm everywhere. It is the studies are going on in all areas. The angiosperm studies are going in one way or other. So we cannot depict the studies in a single phase. This is the studies conducted in Kerala in a single slide. So only so you can imagine how the studies are progressing. Only this much studies in our area. That is from 1983, Vohra et al. Started studies in Silent Valley National Park to 700 species in 2024. Most of the species we have reported, but Dr. Daniels also reported some species from Silent Valley. Then Naya Wildlife Sanctuary, they have published 2 books from this area. So including that species, we have 7 species. So there are some repetitions of species, but still it's a treasure house. So this is our new species. One first book, 2005 book, Bryophytes of Wayanad in Western Ghats. This is my PhD work. And this is our second book in 2017, Bryophytes of Kerala, volume 1. And second volume we are planning. And then another book, Bryophytes of Hanamari National Park. This is one of my PhD student, Mufid's work. Then Phenology of Western Ghats. This is also my first PhD student's work, Manchula. So I have shown you this photograph. This is very interesting. This is Sahyadrika. So you can see the lower photosynthetic region in this species. And very interestingly, we can see that the species grow during May. And at that time, our area, we have flood during this time, June, July, and August. And after that, the dam will be full of water. And after December, the amount of water will be dwindling. Then we can see the area like this. So at that time, we can collect the species with the sporogonium. So up to that period, the plant is immersed. So this is another species we have reported. This is Rixia carolensis. So this is our recent addition. It's from nearby my home. So this is recorded from the human habitation area. So Fissurans Manilaliana, this is also from a human habitation area. So you all are very interested to know what is the difference between Bryophyte and Phenology. So very common species, but it is very confusing. For teachers also, it is very confusing. So Bryum, you can see like this. So there is a cup inside a cup, and it is the sporogonium, the capsule is drooping. And along with this Bryum, you can see in between the hyophylla. So here also you can see some hyophylla. This is the cup inside a cup, young capsule and mature capsule. So this is hyophylla involuta. The sporogonium is very elongated and it is erect. But most of this time, some may collect this hyophylla and confuse it with a Bryum coronatum. So this is hyophylla involuta and the photograph. So this is Phenology hygrometrica. Phenology hygrometrica, here you can see that the capsule is horizontal in position. So You see this. This is hello. Anybody something to say? So I am about to complete. This is my team. I have completed 6 PhDs. Manchula, Deepa, Rajalesh, Prajuda, Mufid and Chandri, and 6 are ongoing. Nishida, Sachida, Sruti, Vinisha, Shakira and Dirti. And some students are doing MSc dissertations also, 3 of my students this year. So they are all doing taxonomic diversity, or along with the taxonomic studies, we are giving some biogeographical studies and some molecular studies also. So thank you very much. So I think
Thank you so much, madam, for your enriching talk. We will now take a few minutes for questions from our participants. Please, participants, feel free to ask your question. Type your question in chat box. Even YouTube participants also can put your questions in chat box.
Somebody has asked a good, is there any field guide for identification of bryophytes? So there is one book on Ganguly's book, that is Mosses of Eastern India and adjacent regions. So with that book, we can identify mosses. So the liverworts also, we have some books that most liverworts by hornworts and liverworts of Khasi and Jainta Hills. So some of our books are also very helpful, and Dulip Daniels also published some books. So, but you cannot identify all species from a single book. So you need different books for identification. We have never described a flora like a gamble.
Madam.
Yes.
Hello.
Yeah.
Madam, I am Suresh from Tamil Nadu.
Yes,
okay.
Yes, good evening.
Good evening, ma'am. How are you, ma'am?
Yeah, fine.
Okay, ma'am. I have one doubt, ma'am. Rixia frutans, already you told that this will not grow in our country. You already said in one paper. So
there is a confusion regarding Rixia flute, and still there is confusion. But we are at present we have kept one collection as Rixia flutans, because we have collected the sporogonium of Rixia flutans only recently. So that specimen with the sporogonium. So it shows the type species, but we have done the molecular studies, but it is unsuccessful, and we are planning to do it along with some Australian workers. So they are also telling the Rixia flutans is very confusing. It is a complex one.
Yes, madam. I collected that one. I discussed it with Christine. Can you understand me, madam?
Yeah.
I am Suresh. Rixia Junagadh and
Rixia. I know.
So I have collected that Rixia flutans. I have seen many sporogonium in the species. So that's why.
But we have also collected Rixia flutans, but it is in marshy area. But that species related with the stricta, then Stenophila. Everything is a complex, and it is very difficult. So we will sort out this soon. So at present we can say, but in our area in Kerala, we could not collect the species with the sporogonium yet. Only recently we could see, but it is also shows sporogonium only when the water dwindle, or in a marshy condition it develop. This aquatic species never produce this sporogonium. So we have never seen, but it doesn't mean that it is not Rixia flutans.
So Rixia flutans, I have collected that one, but also I sequenced from that Rixia flutans. It match with the Chinese person on species.
Okay.
I have seen many sporogonium in the Rexia potency. So
Okay.
that's why I have doubt. Okay, we have to discuss with madam. So
Hmm.
I am waiting, but I
Yeah,
have
sure,
that.
sure. We can discuss later. Okay, no problem.
Okay, ma'am. Okay. Thank you, ma'am. Thank you, ma'am, for your.
Okay, thank you. So
Uh,
my address is here. I am from, yeah, can use net. Some of them asking my address. You can collect from my mail ID and contact, or my number is there. You can use
yes,
it.
madam. Yes, I have that, ma'am. Ma'am, I will send to that Junagadh. So there is no much rain. That's why I'm waiting for collect. So if I collect bin, I will send to you soon.
Sure,
Okay.
sure.
Okay. Thank you, ma'am.
Okay, okay.
Yeah.
Some another from Assam. So how can I contact you, ma'am? So you can mail me, no problem. To follow to identify the bryophytes, what procedure we need to follow to identify. So it is very difficult to say in a single word, what are the procedures to identify the bryophytes. So it needs time.
Yes, and review.
Yeah. What would be the best time to collect liverworts from the Western Ghats? This time, Maharashtra now, this is the best time. So we can collect gametophyte during June, July, August. From September onwards, we can see the sporogonium. So September, October, November, we can collect sporogonium.
Someone asked, have you worked on medicinal properties of bryophytes?
So there is no report of direct economic importance or direct usage of bryophytes, but there are some reports or the studies are going on. So you all know that about the peat moss. For peat moss, from time immemorial, the people are using this species, but also there are some secondary metabolites. So identified in some species from China, and some works are going on. And economically, it is very useful. Horticulture, it is very important. So, and normally I will teach that portion also, but only 1 hour. That's why I have skipped that area. The importance of bryophytes in our area. Horticulturally, it is very important. Ecological importance. So it's very, we cannot imagine the ecological importance. The pollution indicators, the climate change, then most of them are growing with the within bryophytes. It acts as a surface for growing some other spores and some seedlings. So ecologically, it is very important, but the least studied in our area also.
Someone asked, what are the key characteristics of Exormotica ceylonensis and how it is different from Exormotica tuberifera?
So Exomotica ceylonensis and tuberifera, very few difference. In tuberifera, we can see tuber. That's the difference I have seen. The tuberifera we have collected with the tubers. So, but apart from this, there is not that much morphological difference. So I think we have to do molecular studies to differentiate the ceylonensis and tuberifera. So somehow I think we can synonymize with the ceylonensis, this tuberifera also. Other characters are similar. Hornbird species from Western Ghats of, it's very difficult. Hornbirds, I think from NPRA, National, that Dr. A.K. Asthana has completed hornbird studies. His PhD thesis on hornbirds. There is a book on hornbirds of India. So you can use that book, Hornbirds of India, that is Bibliotheca Bryophytorum. Bryophytorum Bibliotheca, that is the publishers. So in that, he has described nearly 40 species of hornbirds. So that book is very useful.
Anyone else to
What
ask? Any-
ecological studies can be conducted for simple mosses present in the wetlands and effective study design. So we have to plan it. So it's very difficult to design design the bryophyte studies. So for ecology. So transects and quadrates. So most of them use 10 into 10 quadrates studies. So wetlands also, some very few species are seen in wetlands also. So what evolution advantage Bryophyte offer in their habitats? Evolutionary advantage. So we all know that the Bryophytes, without Bryophytes, the forest is very shabby, isn't it? So the all Bryophytes are with they are resurrection plants. So when the Bryophytes are seen in a forest, so there is a feel. It gives a microclimate. So that is the evolutionary significance. So I think next speaker has joined. So it is better to stop here. And anyone need any identification or some corrections, so you can contact me. So Bhagyashree knows my number and email ID.
Yes.
Okay,
ma'am.
thank you.
Thank you, madam.
Okay, thank you very much.
So it is now my pleasure to welcome and introduce our second speaker. Hello, sir. I'm audible?
Oh, can you hear me?
Yes, sir.
Okay, I need to go onto shared screen, please. Put my
Yes, sir.
PowerPoint on.
I will give your introduction to each and every participant.
Okay.
Yes, sir. So our esteemed speaker, Professor Jeffrey Graham Duckett. Professor Duckett is globally renowned bryologist, currently serving as scientific associate at the Natural History Museum in London, and he is emeritus professor of botany at Queen Mary University of London. He has extensive academic background with over 250 referred publications and 7 edited books in area ranging from bryophyte, pteridophyte to red algae. His research spanned a wide array of topics including systematics, evolution, desiccation biology, and fungal symbiosis. He is with international field experience across Europe, North America, Africa, Asia, Australia, and South America. Professor Duckett has greatly advanced bryology on a global scale. He was awarded in Linnean Society Medal for Botany in 2008 and Richard Spruce Award in 2007 by the International Association of Bryologists. Recognizing his outstanding research in bryology spanning over 25 years. Professor Duckett has held esteemed role in field, including serving as president of International Association of Bryologists and being elected as honorary member and farm president of the British Bryological Society. It is a privilege to have Professor Duckett with us to share his profound expertise with us today. Please join me in welcoming him to the virtual stage. Please, sir, now you can share your screen and start your talk.
Hello, can you hear me all?
Yes, sir.
Can you see the screen?
Yes, sir.
Can you see? You can, okay. Thank you. So everybody can see there's a visual there, unraveling the complexities of bryophyte phenology. I've chosen to speak about bryophyte phenology because I think it's something we can all identify with and we can all do very easily. The images I've put up are male inflorescences, scanning electron micrographs of Olecomnium palustri and Niam hornum. We're all used to seeing scanning micrographs, leaf surfaces, spores, but we haven't really gone in for looking at other features with the scanning electron microscope. So I'm going to put up quite a few images of sexual bits. So this behind my talk is a new project funded by the British Bryological Society looking into phenology. We only really started this year and we found a lot of things out so far, mainly things we've never looked at before. We've been staggered just how little is actually known. Lots of new questions to pose, which lots of you can identify and do work on very easily. Involving amount of time to dissect the phenology of a particular
Excuse me,
stage.
sir, your screen is not visible, sir. Sorry.
Sorry?
Your screen is not visible, sir. We are not able to
So the
see
screen is freezed.
Has anything happened now?
Maybe you can stop sharing, then you can do it again, sir.
Stop sharing.
Yeah, now do it again, sir.
I'll go to screen. Have you got an image now? Have you now got an image, please? Hello?
Yeah, yes, sir.
Have you got the image now?
No, sir.
No, sir.
Oh, let me try another way.
Yes, sir.
So let me try something else. Let me try another way.
Just share the entire screen, sir. Then it will come. Yeah, yes, sir.
Have you got it now?
Yeah, it is coming, sir. You have to just select that your presentation.
Have you got it now? Have you got the whole thing now? No.
Not right, sir.
Have you got the image now?
No, sir.
Have you got the share now? Have you got the share now?
No, sir.
No, sir.
No. Oh. Let me try. Let me try another way. What's it called?
Sir, just click share, then share screen. Then you just.
I'm trying another way. No, that's not doing it. I'm sorry about this. I don't know how to work this. I'm trying another way. I might
Yes.
do it this way.
Now you can
Let's
choose.
try.
You can just select your presentation, sir. Then it will come. Now I
Hang
can
on,
see
let's
your desktop. You just open your presentation, sir.
No, I'm still trying to find a way of opening. Sorry about this. Sorry. No, I'm still. I can't get the screen to go. It's the right thing to open. Let's try. You got it now?
Yeah. Yeah, it's now.
Have you got the image?
I'll see.
Yes, sir.
Have you got the images yet?
Yeah, yeah.
Yes.
You can see the image.
Yes, sir. Now it's visible.
You should have on screen an image which says unraveling the complexities of bryophyte phenology. Is that correct?
Yeah. Yes, sir.
Okay, and there are two scanning electron micrograph images. Right. Right. Have you got an image that now says New Project 2024, British Biological Society? Have you got that? Say yes, please.
Yeah, yes, sir.
Yes, sir, it's visible.
Yes, sir.
Just by getting.
You've got the image, have you?
Yes, sir. Now it's working. So you can continue.
Okay, that's fine. Okay.
Okay.
Right. It's a new project. And we found a lot of things out this year. A lot of things we never looked at before. We've been staggered just how little is known about phenology. Time involved, it takes about a half an hour to 2 hours to sort out what's happening with the phenology. The sad thing about this, I'm very old. I wish I'd started this a long time ago. So why do you want to study phenology of bryophytes? Well, for a start, it's good to have specimens which have got sporophytes. You know, continuing from the last speaker and talking about Rixia. In some cases, we need fertile specimens for identification, separating dioecious and monoecious species, for example. Also, it's a very useful handle on how climate change might affect bryophyte life cycles. If climate changes, is the way they reproduce going to change? How do droughts affect bryophyte survival? Also, looking at life cycles tells us a lot about periodicity. We can't count tree rings in bryophytes, but we can measure how fast they grow. These are shoots of Polytricum formosum, you know, the genus Polytricum. These are male shoots. All Polytricaceae are dioecious, and each year, at one time of the year, it produces inflorescences, male inflorescences. When all the sperm have been discharged, it starts to grow again. So this year, this arrow here points to the growth in this year, this arrow growth in 2023, 2022, 2021. And you can quite see that 2022, Polytricum formosum grew a long way in Britain, grew less well in 2023. But why do we study phenology? Because we really don't have much idea what's going on. It's very different activity from recording, identifying species, because we can work with common species, and it's a question of what turns you on. If you are dotty about mapping bryophytes or sequencing, then looking at phenology is very, very different from this. If you like to read this, phenology epitomizes cryptogamic botany. Half the time you can't see what you want to see, and when you can see what you want to see, you know you've missed it, so you have to wait another year. Now, for starters, recognizing sex organs in bryophytes. Illustrations in textbooks aren't very good. In fact, one of the best illustrations, I'm sure you're familiar with, is the nice pictures in Parihar's textbook of sex organs in Funaria. So when you see sex organs, you need to be able to distinguish whether they're young or old, male and female. And so I'll show some pictures which illustrate this. These are some pictures. The species don't matter, but what does matter is this is a male inflorescence of Hookeria lucens. These are the antheridia, these sac-like structures, and these things here, these are the discharged spermatozoids. This preparation was a squashed male inflorescence, and you can see the antheridia discharge their sperm. This is an inflorescence of Tortula truncata, synoecious inflorescence. This is a young antheridium here, younger than these here, and these are Archegonium. This is a young Archegonium which is green. This is an old Archegonium which has got a brown neck. Old Archegonia have brown necks. Very nice long Archegonia in Atricum undulatum, long necks, the swollen venter here. An interesting thing about the necks is the necks are sometimes twisted. That's a left-handed one, that's a right-handed one. It's very unusual to get twisting in both directions. Most twisting in bryophytes always goes the same way. For example, peristome twisting is always the same way. An illustration of some mature, post-mature Archegonia, the moss Lytidiadelphus laureus. Archegonia in Pellia. This is a young Archegonium, and here you can see the egg cell, the ventral canal cell joined to it, and these the neck canal cells. This is a mature Archegonium where all the neck canal cells have disintegrated. So, another important feature of bryophyte reproduction is whether they produce their antheridia first or their Archegonia first, protandry versus protogyne. In some cases, they're obviously protandrous, funaria is, tortular truncata is, but a lot of things are protogynous with the females first, which is most of the case in Splachnaceae and in pleurocarps. Another aspect of phenology is paraphyses, which are defined as uniseriate hairs associated with antheridia and Archegonia. Now, it may amaze you to know there has never been a systematic survey of paraphyses. There are no systematic documentation, the number, morphology, presence or absence, let alone their function. Note that the paraphyses are not the same as axillary hairs, where there have been extensive surveys on numbers and morphology. Goebel, way back in 1905, suggested paraphyses were secretory. Our recent observations indicate that this is not the case. We're at a loss to know what paraphyses are doing. And you have some pictures of paraphyses. The most distinctive of all paraphyses are in Niam hornum, multicellular, and the ending pointed tips. And interesting, the male and the females in Niam hornum, this is male with the antheridia, female with the Archegonia here, they're the same. The scanning electron micrograph of the paraphyses, which I think are particularly beautiful, really very easy to get scanning images like these. That was Niam hornum. Niam hornum is unique so far. If you look at other paraphyses, they're very different. The sister genus Rhizonium, the paraphyses aren't pointed, they're rounded, and the males here are different from the females here. Rounded paraphyses are much commoner than pointed ones. This is another example. This is Bryotelia, which has got rounded paraphyses, and this is Philanotis. Another example, and this is common nature of paraphyses in pleurocarps, they are pointed. This male Rytidiadelphus laureus pointed paraphyses, and this is pointed paraphysis in Thuidium. Nice example, Heucheria. You know, the Heucheriaceae have very large leaf cells. They also have very large cell paraphyses, huge cells, both same male and female, these huge cells. At the other extreme, you get paraphyses in Andrea, which are extremely thick-walled. These are the paraphyses in Andrea. These are the axillary mucilage hairs, which are completely different morphologically. The Polytrichales have interesting paraphyses in that they are not filamentous, but they are flattened plates. This is a young paraphysis, another in Polytrichum, and these are the axillary hairs, which are very different from the paraphyses. And the scanning electron micrograph showing Polytrichalian paraphyses are flat plate-like structures. Here, by contrast to paraphyses, are mucilage hairs. This is in Diaphysium. And what happens, these filaments secrete mucilage. And what you can see here, these little collars, these are where the cuticle has broken along the filament and the mucilage has been released here. And the broken cuticle you can see. So it's a particularly nice example of mucilage hairs. Paraphyses in liverworts are very rare and they integrate with mucilage hairs. Mucilage papillae are much more widespread than Paraphyses in liverworts. Some liverwort Paraphyses. This is in Scapania gracilis. These are the mucilage hairs here, which are very different from these much branched and sort of plate-like Paraphyses. Another important thing to notice with sexual reproduction in bryophytes is calyptras. Calyptras are ubiquitous mosses and liverworts and in hornworts. They provide embryo protection. And this is one of the key features which you rarely read about, is that it's a key feature separating bryophytes from pteridophytes. Embryos are not protected in pteridophytes. And calyptras persist right up until sporophyte maturity in many bryophytes, particularly sphagnum. In mosses, we see considerable elaborations of the calyptra, which develop from the young embryo stage. For example, the orthotrichales, as you all know, have got hairy calyptras, as do the polytrichales. Hairy calyptra are useful in taxonomy, for example, separating species of Eulota. Amazingly, I couldn't find any decent illustrations of the just scruffy sketches in Polytrichum. And to our great surprise is the calyptra of Polytrichales resembles the protonema, the young stages. This is a young calyptral stage in Polytrichum, thick-walled when they're old, but in the beginning they mirror protonema, use the axial filaments with side branches, and the side branches coil round the main axes. And if you look at the scanning electron micrographs of calyptras in Polytrichum, you're in for a visual feast. Well, I think that's pretty anyhow. They're absolutely fantastic images. Anyhow, we'll shortly be publishing these to get them into the public domain. You think that a good place to start looking at bryophyte phenology is Janice Glime's encyclopedia for the Bryophyte Bible. Remarkably, Janice Glime's book doesn't give very much information on phenology, no general conclusions. I think that it's influenced by short and long days, light intensity, nutrients, but most of the information in Glime is obtained from cultured plants, and we have very little good information on how on bryophytes in nature. One thing is, let's start off with mosses. One thing about mosses is interesting is that mosses, the sporophytes are very persistent, so you can see them at long periods of the year. Liverworts, perianths are persistent, but not sporophytes, which are highly transient. In hornworts, there's continuous growth of sporophytes. So sound information on phenology is one of the biggest gaps in bryophyte biology. These are my own observations on when you can see capsules. In sphagnum, I only see them in the summer. In Andrea, I see them all the year round. A few species, you see them in the autumn and the winter. But in many common bryophyte species, you see capsules all the year round. But this doesn't really help us in unraveling the development. Big players in the reproductive cycles of mosses was Stanley Green, who defined stages in sporophytic development. This was refined by Lloyd Stark, who identified 4 gametangel and 8 sporophytic stages in moss sporophyte maturation. And then we looked at and divided these up into 12 stages, and that's published in Bryophyte Diversity and Evolution. Perennial mosses seem to fall into 2 groups. Autumn gametangia, sporophytes maturing the following year or later the same year. Spring gametangia, sporophytes maturing in the same year. And you follow common mosses through their life cycle. This is Bryum capillari, produces, these are male shoots, which you can see in the spring. By late spring, May, you get seta, young seta, and then you can follow the maturation over several months in the summer from expanding capsules to long dehisced capsules here. And then you can document all these. This is all published when you can see the gametangia in the spring, when you can see fertilized eggs and young embryos, when meiosis occurs. So you can get all the different stages. And this is very, very easy to do. All you have to do out is go and sample and look at bryophytes going on your doorstep. And I'm sure that probably the first bryophyte you look at growing on your doorstep, we know nothing about this sort of information. Polytrichum is interesting in that polytrichum has a 16-month cycle from gametogenesis to spore discharge. If you think about flowering plants, Bryophytes, they normally have an annual cycle. Well, polytrichum is extremely odd in that it only has a 16-month cycle, which is very odd. I'll give you some examples of the polytrichum life cycle. These are the sporophytes. These are young sporophytes, the spear-shaped stage, and these are dehiscing sporophytes when they're shedding the calyptra, and these are older ones which have shed their calyptra and are discharging spores. These are some like young capsules here. These are old male plants. So we've looked at some mosses in detail. For example, Smith in his British Flora describes capsules late autumn to spring in Heucheria. What we now know is they produce their sex organs in the middle of the winter. These are mature sperm in January. This is a paraphysis, whereas throughout the summer months, all you have are young embryos. This is an old archegonium in the summer. This is a young embryo, and this is, if you squash these, you get out the embryos, and you can do that very easily. And this is the base of this is the foot. This is where the sporophyte obtains its nutrition from the gametophyte, and these are highly specialized transfer cells. You can actually see these with the light microscope. So Heucheria has sex in the winter, and then it sits throughout the summer as a younger embryo stage, dormant through the summer months, and the sporophytes mature in the winter. Spores to spores, it's only half the story for phenology. We don't know how long it takes for a spore to be established to when the same plant produces its sex organs. So we haven't much idea. Fires are a very good starting point for looking at when species start. And we've actually looked at the phenology of fire, of what happens on fire sites, Funaria versus Ceratodon, and then more perennial is Polytrichum. If you get arable fields near you, it would be a great exercise to go and look at how long it takes from the bryophytes to come up to when the arable field bryophytes produce spores. We really do not know. Another interesting aspect of bryophytes are sex ratios. We assume if in dioecious species it's 1-to-1, but very little definitive information. We now know what happens in Marcantia, as I'll show you, and Ceratodon. Edinus and Bissang looked at a lot of mosses and liverworts, and they discovered a distinct female bias. But phenology in relation to sex ratios with little idea. When it comes to fires, what we know after fires, Funaria comes in first, then Ceratodon, then Polytrichum takes over. Examples of Ceratodon, most of the year, what you see in Ceratodon is this, old dehisced capsules. In the autumn, however, it looks a bit different. In the autumn, these green plants are female, pointed here, these brown ones, these are the males. There's always an excess of females over males in Ceratodon. And I put this picture in because at the spear stage, you get water droplets after rain on the Ceratodon. I just think sphagnum, in order to see gametangia the gametangia produced in the winter, and you don't see anything until late spring, early summer, when the capsules become apparent, and then a month later the capsules are all dehisced and then they disappear. If you want to look at sex organs in sphagnum, you have to go in the summer. Now, I've been interested in all groups of bryophytes equally. I do different things with different ones, and I couldn't miss out on hornworts. In the Northern Hemisphere, well, a lot of hornworts are ephemeral, and they just produce sex organs continuously, and sporophyte development is continuous. However, in the tropics and the Southern Hemisphere, hornworts are very clearly seasonal. This is the biggest of the hornworts, Northothorus giganteus. And I've seen this several times in New Zealand. This produces its sporophytes in the spring. Like Northothorus, at other times of the year, you don't see sporophytes. And I think I have to illustrate, this must be my favorite bryophyte locality in the world. This is a cliff made up of glacial drift from the last glaciation. As you can see, there's some scale objects here of humans. It's a huge wall, and there are 5 different hornwort species growing on this wall. The human touch. This is a picture of the wall with Chris Cargill and Nancy Slack, who some of you must have known Nancy and Chris Cargill, sampling the hornworts. And this is Phynomygestrus urticalix, this type locality. And you can clearly see the new thalli are not producing sporophytes as the old ones are. This is clearly seasonal. I have a need to go back to New Zealand and look some at different times of the year. But it's an ideal thing for New Zealand bryologists to do, look at seasonality in their hornworts. Now, liverworts, these are some examples of sporophyte production from Gene Payton's book on the British and Irish bryophytes. And interesting here is that some liverworts seem to produce sporophytes all the year round, where some are much more seasonal. This is not what we have found. Payton, I think, is inconsistent. She talks about presence of perianths, but not once inside them. Seeing perianths is not the same as seeing sporophytes, as I will illustrate. We know so little about phenology in liverworts that nobody's even defined the different stages in development. Antheridia, when they develop and mature, pre-perianth stages, long persistent perianths, swollen ventres with young embryos. There's another stage in liverworts which we know very little about, and that some liverworts show arrested meiosis. And how long it takes the spores to mature is short. The capsule dehiscence is a very fast process, unlike mosses. What we have looked in great detail is Marcantia. And to our surprise, we find that Marcantia, this is Marcantia polymorpha. And I think it'd be interesting to know what happens in other Marcantia species. Marcantia polymorpha, the model Marcantia in the wild, can produce up to 3 generations of Carpocephala. What did surprise us was there's asynchrony. The Antheridia are produced earlier than the Archegoniaphores, and we really see a sex ratio 1:1 only in the summer. The other amazing thing about Marcantia and a lot of liverworts is they have an almost 100% fertilization rate. Sexual reproduction in liverworts is incredibly efficient. Huge fertilization distances. So I wouldn't think it was motile sperm, but sex is very efficient in Marcantia. Some pictures of Marcantia because they're pretty. Germinating spores produce highly gamiferous filament thalli first. Then first we get males. These are young males. Then young males and old males, and it's only later in the season that we get Archegoniaphores. These are mature males, mature Archegoniaphores. And then later in the season, you see other generations of females. You've got young females and old females, and you're getting several generations per year. And here we have old females and young males. And something I've got to show you. You never expected to see a picture of railway engines, did you, on a bryophyte presentation? Well, the reason for this is that railways and steam train areas turn out to be incredibly good areas for Marcantia. There are square meters of Marcantia growing here. And I went first of all in the spring and got a great shock. The only Marcantia I could find right next to these railway lines were females. So you looked at Marcantia in the summer, you'd think it was purely female. This is in the summer, this is in the autumn, this is the winter. You have to wait for the winter to see the males. Our leafy liverwort Frulania, which is said to use sporophytes all year round. This is Jill Carroll, admiring Frulania tamarissai dilatata, Frulania dilatata in the winter. And what you can see inside the perianth, you can see these black, almost mature sporophytes, which mature during the winter only. The perianths persist all the year round, but you'll only find the sporophytes produced in the winter. These are young antheridia in the winter. This is an antheridium. This is a typical archegonium, which is post-mature in the early spring. And throughout the summer, inside the perianths of Frulania, things look like this. This is a young embryo. This is the archegonial neck. One plant that really has captured our imagination is Cephalosia divaricata. This is because we've got some money to look at reproductive, the underground, what's happening underground, the heathland microbiomes in Cephalosiella. Cephalosiella divaricata shares the same fungus as the Ericaceae on heathlands. And so we got very interested in looking at the reproductive biology of Cephalosia divaricata. The only previously published information was Peyton that says you can see perianths all the year round. So you can. This is Sylvia Pressel looking at perianths in the summer. This is Sylvia Pressel looking at perianths in the laboratory. What happens in Cephalosiella divaricata is not what we expected at all. You can see perianths all the year round, but you only see young sex organs, young females in the winter. These are young archegonia. From the winter onwards, we see young embryos. So throughout the summer months, when Cephalosiella is very often dry, you just get young embryos. Scanning Electron micrographs, this is a male shoot you should see in the winter. There's an antheridium. This is a young female before it's produced. It's perianth, full perianth. And interesting, the male shoots are interesting. They produce antheridia in the winter, then they switch to gemma production. And this is a production, like a male plant which is producing gemma in the spring and throughout the year. So Cephalosiella is clearly highly seasonal. And so we're shortly producing the, publishing this, which is completely unexpected and really rather good. Another plant we hadn't a clue about was Andrea. Or Spring said that you could get capsules in Andrea, spring and summer. Well, I knew this couldn't be right because I lived in North Wales for 9 years, and every time I went out collecting bryophytes throughout the year, I always saw sporophytes. Anyhow, we started looking in more detail at Andrea, and the update on Andrea is that Andrea produces its sex organs in the winter. These are young antheridia in the winter. These are young archegonia in the winter. After, in the winter, it produces young embryos which stay as young embryos. This is a young embryo in the middle of the summer, and this is a fertilized archegonium with a young embryo. This is a dissected out embryo. So throughout the summer, it's sitting there with young embryos, and the sporophytes only mature in the winter. So we've now unraveled what's happening in Cephalosiella hookeria and Andrea. Sex in the spring, sporophyte maturation late autumn, winter. And our conclusions so far, we've only found one perennial moss with no seasonality whatsoever, and that's Cryphea. There are two groups of mosses, winter Gametangia, autumn sporophytes, or autumn Gametangia, spring sporophytes. This is an interesting phenomenon, the alternation between gemi and antheridia on male plants. Big question is, why are some species autumn? Why are others spring? We do not know. Anyhow, to unravel phenology, the key stage you require are young archegonia and antheridia. Once you've got that, you've got your starting point. You can work out how long it takes from seeing the sex organs to fertilization to spore release. Most species are around 9 months, except in the Polytrichales, it's 18 months. I'll stop there and stop the screen share, I hope. And go back to everybody. Okay.
Thank you.
Hello.
Thank you, sir.
Right. I hope that was of interest to people.
Thank you, Professor Duckett, for your insightful presentation about phenology of such a tiny plants. We will now open the floor for questions from audience. Who are participating in YouTube platform, they also put the questions in as a comments.
So I haven't looked at the chats. I'll look at the chats in a second. I don't think I've got any chats. Somebody asked the speaker, a previous speaker. Sorry.
No, go ahead, sir. Go ahead. Some questions are there in YouTube that I will put in the comment box, sir.
Somebody said Pulkit asked a question. Well, Pulkit asked a question about symbiotic associations. I can answer that one because that's what we work on. Can you hear me? Oh, yes. You can hear me. Yes. An interesting thing. There are no symbiotic fungi in mosses. Mosses do not do symbiosis. Liverworts, the hornworts do. And the symbionts are very interesting that in Bryophytes you see some of the most primitive symbiotic fungi in the world. A marvelous system for looking at symbiosis. And as well as fungal symbiosis, there are blue-green cyanobacterial symbioses ubiquitous in hornworts and also in the Blasiales.
Hello, sir.
Hello.
I have one doubt, sir. Yes, sir. Sir, I am continuously identifying about Rixia. How to identify easily with Rixia? Do you have any idea about it?
Sorry. Could you repeat that, please?
Sir, I am researching in Rixia. I have doubt from that one. Any easy identification for Rixia? Do you have any idea about it?
Mm-hmm. Sorry, I can't. I do apologize, but I can't tell what you were saying.
I am researching in Rixia.
Yes.
How to identify in Rixia based on the morphology character?
Mm-hmm.
Is it easy to identify
Rixia.
that Rixia by morphology character?
Mm-hmm. Sorry. So you have to. I think to identify Rixias, you need to. You need to find the best taxonomic accounts. Rixia is interesting from the point of view of reproducing of phenology because some Rixias are perennial and some are and only survive from sport from one year to the next via spores, and others are perennial. The thalli can dry out and rehydrate. It's very interesting. Sort of chain differences in biology, and that would be a fascinating thing to do if you live in a nice seasonal climate where you've got Rixias on your doorstep, to see which survive and which don't. Somebody asked.
That's the main problem to identify Rixia based on the morphology character. It's a difficult one. I struggled many times for when I see the Rixia. So, do you have any simple idea for that identification of in the Rixia? Only.
No, sorry, I'm very sorry, but I can't help. Somebody just asked. You saw my pictures. The pictures in my presentation. They were all fresh material mounted in water. You can also make them semi-permanent, but using any of the permanent mountains. What I would suggest, if you want to make permanent preparations of Antheridium archigonia, I suggest you dissect out the preparation first, then you fix the material. You can do it in aldehyde or in 10% alcohol for a couple of hours, and then put it in the dehydrating in the permanent mountains. But doing the fixing first will preserve cell structure much better. Okay.
Okay, thank you, sir.
Sir, someone asked in chat box. I was wondering if the microscope slide of Antheridia and Archigonia section are semi-permanent or permanent preparation. Additionally, could you please share the method used to prepare the slides?
Okay, sorry. Yeah, they weren't permanent. No, the permanent record are the pictures you saw. And the permanent record is the herbariums. The permanent record, if you like, are the pictures I've shown plus the herbarium specimen. Yeah, we always have a good practice. The key thing is to have a herbarium specimen. Anything you're going to record and do research on, the best practice has to be you have herbarium specimens. Okay, as I said, you could make permanent preps, but so for the phenology stuff, there's nothing nicer than actually looking at fresh material. It's also better to use fresh material than looking at rehydrated herbarium material. So this is so doing phenology really is a case of going out in the field and collecting fresh material. You don't have to go very far. That's the nice thing.
Someone put the question in the YouTube platform. Which microbes are found associated with bryophytes?
Basically, the same as are associated with just about every land plant. There's a huge microbiome of bacteria, fungi, and cyanobacteria. So There are quite a few papers now. If you want to Google Bryophyte microbiomes, you'll come up with quite a lot of papers on these. But we know that sort of microbiomes in general. What we don't know about are different microbiomes. Whether, for example, the microbiome of sphagnum is different from the microbiome of mosses growing in an arable field. There be some things in common, but nobody's really set about characterizing the different microbiomes. I mean, this is one of the things we're doing our research on now, are the microbiomes, especially the fungi associated with heathland bryophytes, because heathlands nitrification is a huge problem affecting heathlands, certainly over large areas of Europe. I would imagine nitrification is also a problem over large swathes of India, and we're doing experiments adding nitrogen to see how that affects the bryophyte microbiomes. Okay.
Dr. Anuj Saxena asks whether antimicrobial compounds and phenology of bryophyte has any relation.
We don't. Right. We don't know. We don't know. However, if you look at bryophytes under the microscope, one thing you're very struck by, unless it's old necrotic material, one is very struck by the absence of microbes growing over the surface of bryophytes. You can go out and collect a bryophyte and look at the leaves. You don't see microbes. You don't see microbes very much at all. Whereas if you go underground and start and look at moss rhizoids, for example, they're covered with microbes. So it suggests there is something about the above ground bits of bryophytes that are highly antimicrobial. But I mean, which again is very interesting. Okay.
Thank you so much, sir.
Pleasure.
Now I invite our Director of CWCS, Dr. Nishit Dharaya, for a formal vote of thanks.
Namaste and very good evening from India. Good afternoon in the UK and Europe, and maybe the morning in the United States. First of all, it was a wonderful interactive session. Both the speakers have given their best, and I must congratulate our team at CWCS, Center of Excellence for Wildlife and Conservation Studies in Bhakt Kavi Narsi Mehta University. I must congratulate my team to organize such a nice event. Friends, I am a zoologist. I don't know much about botany and especially these lower plants, but I learned from Bhagyashri that these minor plants or minute species of plant called bryophytes, they are actually the green threads of the biodiversity. They are holding the soil, they are early successional species, and they play a very vital role in reviving any ecosystem. And that is why these are very important plants of the plant kingdom that affects the entire wildlife. So at this juncture, first of all, I would like to thank both our eminent speakers who are the leading bryologists of the world, Dr. Manju and Dr. Jeff. Sir and Madam, you have given your valuable time for our center, and you have delivered a very scientific and very lucid talk, and that has enriched our knowledge about the bryophytes and the role of the bryophytes in the ecosystem. So on behalf of Bhakt Kavi Narasimha Mehta University and our center, I would like to thank you very much, sir, and very much, madam.
Thank you. Thank you very much.
Yeah,
Thank you.
I would also like to thank Dr. Rajesh Ravia. He is a botanist and he is the member of our advisory committee of this wildlife center. It was his idea to organize this World Bryophyte Day, and it was really a wonderful idea, and I welcome this idea. So I would like to thank Dr. Rajesh Ravia on this juncture.
Thank you, Nishi sir. Thank you.
Yeah, my big thanks goes to Rani. Rani is Bhagyashri. She is a junior research assistant at our center, and she is, I think, youngest bryologist who is appointed on the IUCN SSC Bryophyte Specialist Group. She handled and she organized this entire program very nicely. So I must thank Bhagyashri for organizing this very interactive scientific event as a webinar. First, finally, I would like to thank all the participants because this entire webinar is not only on the Zoom, but it is also live on our Facebook and our YouTube channel. So people from 18 different countries and 26 states of India, they are either botanist or conservation biologist or a bryologist. They are all have taken benefit of these two eminent lectures. So I thank all the participants. And I wish that you will keep joining us, you will keep with us, you will be with us in all our future programs. Because the Center of Excellence for Wildlife and Conservation Studies is the main objective of this center is to aware the people about the biodiversity, the role of biodiversity, and especially these lower plant and animal taxa, which are lesser known, which are understudied, and their role. What is their role into the ecosystem? So we'll keep organizing these kind of events. We are also planning to organize the offline workshop on the bryology, where we will take you out in the field. We will help you. We will show you how to collect the bryophyte sample, and then Bhagyashri and our experts will show you how to identify these bryophytes and how to study them in the ecosystem. So this is our upcoming program. So I'm sure that you can join in this upcoming program of our center, and you will stay with us like this for every event that we organize at our center. So once again, I would like to thank everyone, and above all, I would like to thank our Vice Chancellor, Professor Dr. Chetan Trivedi, who has approved this event and who has allowed us, given the permission to organize this event on the World Bryophyte Day. So once again, thank you everyone. And I would request all of you to on your camera so we can have a group photo. Thank you guys for starting your camera. And I will ask you guys to smile. So, yeah, so I can take a picture. Still, a few people have not on your camera, so I'm waiting for them. Thank you. Thank you very much. Still, I am waiting for a few people. I'm still waiting for a few more people. Utkarsh, you can come this place for the group photo. Yeah. Smile everyone. Smile for the Bryophytes. Thank you.
Okay, thank you. Thank you very much.
Thank you very much. You guys can leave now. Madam and sir, can you please be here for a while?
Sure.
Dr. Jeff and Dr. Manju. Dr. Rajesh, Dr. Manju and Dr. Jeff, please be here.
Yeah.
And the participants can leave now. Thank you, everyone, and have a great day. Yeah, very soon we will send the e-certificate of participation in this webinar to everyone. Namaste, madam.
Namaste.
Hello, Dr. Jeff.
Hello.